Hello, class. Hope you are ready for another lecture. We just finished talking about cell division, mitosis, meiosis, and all of that is moving the DNA from one cell to another, right? Or copying it or recombining it. But we haven't talked about what that DNA is coding for and what it's doing. So we are moving on to chapter 8, where we are going to be discussing genetics. So this gets into some stuff that you may be familiar with or remember from other biology classes, but if not, we're going to start right from the beginning so everybody can catch up. So get your pens ready and your coffee, and let's get started. Okay, so we're talking about genes and inheritance. This is actually a picture of the Twin Festival from Twinsburg, Ohio, which is not far from where I grew up, and they have it every year, although I've never actually attended it. And you get a prize for being like the twiniest twin. So things we're going to be discussing in this chapter: heredity, um, genetics, like some of the probabilities, but also chance, the difference between phenotypes and genotypes, and then also gene linkage. So heredity. Um, Most of you have probably heard of Gregor Mendel. He was a Czech monk in the mid 19th century, so the 1800s, who conducted experiments on pea plants to try to understand inheritance. Um, Before Mendel started his work, people really thought that offspring kind of had a blend of their parents' traits. But that would mean that you would always be like exactly in between your parents in height, and your hair color would be exactly between them, right? It would just be this mix. And as we know now, that's not true. Um, Mendel did his work on pea plants, which actually was just a stroke of luck on his part, because a lot of the things in pea plants are inherited in a very discrete manner, and they follow all the rules. And there, as we're going to discuss after we talk about the rules, there are a lot of things that don't follow those rules. So he just got super lucky. But his luck ended up benefiting all of us because he really figured out how basic inheritance worked. So the things that he looked at 7 different traits, and they were all discrete traits, which means that it's an either, everything was an either or. So when he was looking at the seeds, they were either gray and round or white and wrinkled. There wasn't like this whole range of like grayish white and slightly wrinkled, right? It was either one way or the other. When he opened up those seeds, they were either yellow inside, right, or they were green. The flowers are either white or they're purple, right? There's no like purplish white or right or striped or things like that, right? It was just one or the other. So he looked at these 7 traits that are discrete. Discontinuous is the opposite of continuous, right? So he wasn't looking at traits that have a range. And what he did was he would, if he was trying to create, to figure out the genetics of the white flowers, he would keep breeding white flowers with each other until every time he bred them, he only got white. Nothing else ever showed up, right? And he would keep breeding purple flowers with each other until every single time he bred them, they'd always have purple. There wouldn't be any white ones showing up in the every now and then. So he bred these true strains, and then he could mix them and see what happens when you mix a true purple flower with a true white flower, or a true, green seed with a true yellow seed. The good thing about peas is that if you're a good gardener, you can grow them quickly, and it's easy to pollinate them yourself. So he, instead of leaving them outside for like bees to pollinate, he would actually, take the pollen from one and pollinate the other. And as we said, he picked things that are, have only 2 variants. So there wasn't, he could really keep everything under control. Okay, so what did he find? If we're just looking at flower color, and he had these true purple flowers and these true white flowers. And when we're talking about the first time you breed them, this is the parent generation. So this is P. Okay, so he bred purple with white, and the offspring are always called F. So F1 is the first generation of offspring. So in the F1, 100% were purple. Okay, and then he's going to breed these with themselves. Okay, so F1 is going to breed with other F1. He's going to take their offspring, breed them with their offspring. that gives you F2. Okay, so the next generation of offspring is F2. So he breeds F1 with each other, and you get F2. And what did he find? Here we have about 75% purple and 25% white. Why is this? Well, you guys may remember Punnett squares from a previous biology class. So to do this, we're going to come up with just letters for them. Okay, so let's say a, a big P means a purple flower, okay? And a little p is a white flower. And I didn't choose the best letter here because a big P and a little p look the same, but we'll try to keep them straight. I'll put a little line under the little p's, okay? So if we're breeding a pure purple flower, that means it turns out what he didn't realize is that you have one gene from your mom, one gene from your dad, right? And the true purples had both of them purple, okay? The white ones had one from their mom, which was white, and one from their dad, which was white. So here we're looking at the parent generation. And if we breed them, you're just looking at, okay, when they divide during meiosis, what's going to get passed down to the offspring? Well, some of the eggs or sperm would have a big P from one parent, and that would mix with a little p from another, right? For this parent is on the right side here, is going to give a big P again. All they have to give their offspring is big P's, and all this one has to give their offspring are little p's, okay? So what we're looking at here is the offspring. This is F1. I'll circle in purple. These are the F1 generation. All of them are going to look purple because they have that big P, but they're carrying that white little p, right? So then when we do F2, we're going to end up with, we're going to breed the F1s with each other, okay? So we have a big P, little p, is this, like all of them, right? We'll breed these two individuals together, set it up. What can they donate to their children? They could give a big P or a little p, right? We make our square. And you just take one from each parent, right? So this parent's going to give a big P here. This parent's going to give a little p. The parent on the left side is going to give a little p. The parent on top is going to give a big P. Okay, so what do we have in this F2 generation? I'm going to circle in blue all the ones that are purple. Anybody who has a big P is purple. And if you don't have a big P and you only have little p's, you're white. Okay, so big P, if you have even one big P, you end up purple. That means that it's dominant, okay? If you even have one of them, you get it. The little p in this case is recessive. So individuals who have a recessive trait, it's only going to show up if they have two copies of that trait. If they have one big P, it's going to look purple even if they're carrying that white gene. That white gene is hidden, right? Which is why in this generation on the left here that I'm circling again, everybody looked purple even though they all were carrying this little p. So I have some of these terms written down here for you guys. If the two alleles that they have, so when we're talking about these p's, these are alleles. So the trait here is flower color, right? That we're talking about. The p's are the gene is the DNA that codes for that trait, okay? And alleles are different versions of the same gene. So if we're talking about the gene for flower color, there are two alleles. There's a purple allele and there's a white allele. They're just different versions of the same gene. If you have two of the same, if you have either two big P's or two little p's, you're homozygous. Homo means same. So in this case, that could be big P, or little p, little p. If your two are different, you're heterozygous. Hetero means other, okay? So that would be big P, little p. The trait that we observe depends on both copies, right? So someone who's homozygous, you're going to see exactly what alleles they have. But someone who's heterozygous, if you see someone who's purple or a flower who's purple, you don't know. Are they a homozygote dominant, right? They have two dominant genes. Or are they a heterozygote? When you're seeing a purple trait, you don't know that. But when you see the recessive trait, you know, oh, this has to be homozygous. They must be two little p's or they wouldn't look white. So this brings us to Mendel's law of segregation, which is that they segregate, they separate. During meiosis, these alleles are going to separate from each other. So a parent, right, everybody has two copies of an allele. We're diploid, right? We already discussed what that means. You have two copies of every chromosome, but during meiosis, they split and you give only one copy of each, right? So that's that they get segregated during meiosis, right? You have two copies, but only one goes into each egg or sperm. And then, so that's where you can see, when you're looking at the egg or sperm, you can see what the actual genes that this individual has, what alleles are they carrying, because when you're just looking at them, anything that's dominant is going to mask or cover up the recessive one. So if we're looking at here, they're showing the color of the pea. Yellow is dominant over green, so they have big Y's and little y's, right? So an individual here, these two that I'm circling, these are heterozygotes, right? So big Y, little y is a hetero. And you can't tell when you see a yellow pea, you don't know whether it's a big Y like this one, or big Y, little y like these other two. So this is, again, just showing you how things get segregated in meiosis. You already saw this in the last chapter. So when we're talking about these, and I've already used these terms a little bit for you guys, dominant and recessive, right? So the dominant trait, looking at the bottom here, is the one that's visible. If you have even one copy of it, of the dominant trait, it's visible, okay? So this could be, if we're talking about the letter Y for the trait, it could be this, or it could be that, right? The recessive trait, so this is a homozygote dominant, and this is a heterozygote. A heterozygote always has the dominant and the recessive, okay? Or if somebody is recessive and they show that trait, they have to be homozygous recessive. So that would be two little y's. So when we're looking at the homozygous dominant and the heterozygous, so these two, they both are going to look yellow, right? That was the dominant trait. That is their phenotype up here. The phenotype is what you look like, the outward appearance. So it's a purple flower, or your phenotype could be a white flower, or your phenotype could be a yellow pea, or the phenotype is a green pea. The genotype, and it has the word gene right in it there, right, is the actual alleles that they have. So while the two that I have circled down here in blue, this homozygous dominant and this heterozygote, their phenotypes are the same. Their phenotypes are both yellow. Their genotypes are different. Their genotypes are homozygous dominant and heterozygous, right? The homozygous dominant is two big Y's. They're homo, they're the same, and they're both big Y's. That's why they have the dominant trait. The heterozygote has a big Y and a little y. They have different alleles, but they look yellow because they have that dominant one. And we figure out what the offspring, the chances of the offspring looking like something, based on that Punnett square, which is what we were doing when we set it up like this, right? You make a square and then you figure out what the potential alleles that the parents could pass on to their offspring. Okay? I think we're going to stop here just because that was a lot of information. And then we'll start up in the next lecture doing some practice Punnett squares. Okay? So I will see you all in the next